So I was briefed just before, so to present a bit myself, as I'm a physicist by training and a physiologist by art, and now I'm trying to develop a kind of social physiology. And we have heard this morning that cognition is multiscale and we have to take that seriously. And I like to do this kind of joke because, so in Science in 2012, Christoph Kohr described that in an abstract, so he's like the head of the Allen Brain Institute. Biological systems are characterized by a large number of highly heterogeneous components, be they genes, proteins, cells. These components interact causally in myriad of ways across a very large spectrum of space-time, from nanometer to meters and from microseconds to years. A complete understanding of these systems demands a large fraction of these interactions to be experimentally or computationally probed, and he closed the abstract by, this is very difficult. And well, that's where we are. It's very difficult, but as we've seen this morning, multiscale account of cognition and in biology is not new, actually. And so in a country far away, where I come from, France, there was like a guy called Claude Bernard, who basically, damned, I missed my super nice Star Wars video. Anyway, so the point of Claude Bernard was that basically physiology is about linking those different scales and understanding life through systems and through function. And instead of looking at in a reductionist way, as we saw earlier with the talk of Lawrence, because, for instance, people would say, okay, it's multiscale, so let's just link the gene for language or the gene for whatsoever function. Well, the point is that nature doesn't care about those scales, and having a linear thinking about multiscale is not also productive. And there are causality that goes upward and downward between these different scales. And so systems biology has tried to tackle that. And the problem that we have is, we were discussing that, and I may talk again about that in the other presentation about open science, is that cognitive science has been spread all over the disciplines. So here is like a network of the different scientific domain and how they are citing each other. It's like the credibility in science is the currency, apparently. So when you try to find out where cognitive science is, you can find physicists, geneticists, economists, etc. And meanwhile, there's also school of thought. So we are spread from disciplines, and we are spread also by account of what cognition can be. So for instance, I choose 3 examples. Using these myths of the elephant and the blind man, so where the elephant in the room is cognition, and where the blind man's trying to make sense of this elephant. And while cognitivism is kind of falling those days, Bayesian is on the top of the hype, and I tend to define myself as a dynamicist, try to steering the elephant by the tail. And the question that animates me is to realize how separating what is unified initially create paradox. And so, for instance, in the case of social cognition, we have 2 different literature dealing with more metacognitive representational aspects of social cognition, and others who are more in mirroring and sensory motor coupling. And my point is that by separating those scales, we have created this paradox that we need representation of others to interact with them, but meanwhile, developmental psychologists would tell you, you need to interact with others to form this representation. So it's like a chicken-egg kind of issue. And so the point is to go from this systems biology account that try to link scales within individual to a social dimension, because we are part of a social system as well. And for that, as a disclaimer, we are all biased by our readings. So I was biased by my readings of Maturana and Varela. I initially took autopoiesis as a framework for gluing those scales. So for those who are not familiar with autopoiesis, it's a theory originally for biology about autonomy and coupling about cells. And the beauty of this framework is that when you account of cellular dynamics in that way, so as a closed loop that maintains itself autonomously, in a dynamic environment, through evolutionary stabilization of this coupling with the environment, you can see how cells are coupled between each other and iterating from that, so it's like more in action in that case. You can create a metacellular organism with the nervous system or immune system that stabilize not only the identity at the physical level, but also in a form of identity at the informational level. And the thread leads to social interaction as a coupling through sensory motor perception action loop. And so why hyperscanning? Because this sensory motor coupling between each others through development and through our daily life paradoxically has been broadly ignored. Now it's started to kick in, but 10 years ago it was vastly difficult to find papers in this kind of dark spot where two individuals are engaged in dynamic interaction. So like the literature in neuroscience was mostly focused paradoxically on individual isolated from any interaction. And so over 10 years now, I have tried to operationalize the naturalization of social interaction through a threefold program linking hyperscanning EEG from the experimental sites to computational model of dyadic interaction in silico, and more recently, human dynamic clamp, which is a human-machine interface where a human is coupled in real time with a computational model. So that's the simplified emoji version of the Venn diagram. So I'm gonna focus more on the hyperscanning today, but we can speak more about other aspects later on. So at the history level, first disclaimer, I won't speak about Hyperscan, which is the most dramatic game console in history of video games, which is like a Mattel console. And so if you have a Google Scholar alarm for Hyperscan, you have all these eBay stuff about that. I won't speak about it. No, I'm going to speak about hyperscanning as Reed Montag coined it in 2002 in a no-image paper, which explained basically that when you have enough money to have two fMRI scanner, you can do two people simultaneously through internet, which sounds like obvious like that, but technically it's kind of challenging, and you need two fMRIs. So. But the term coin hyperscanning was for fMRI in the first place. So when we say hyperscanning EEG, it's kind of weird because we are not scanning people per se, but that's where it comes from. And so, well, I think I have to deal with the videos to not work, but so it goes even further initially by saying it's not only two fMRI. If you have 10 fMRI, you can also do it. We're gonna add to that later. But what's interesting already in his paper and make it very seminal is that not only he coined the term and described how we can record two people and how it's interesting for analyzing social interaction and social exchange, but also speak about things that are not yet realized, actually. So that's really particularly visionary, because it talk also in this paper about neurofeedback at the dyadic level. So we are not reached that yet, but it's good to have in mind. And so if you look at the number of paper in PubMed with hyperscanning, you see that it's kind of indeed increasing over the last year. And I put like this, like that, because in 1965, actually, there was like the first hyperscanning paper, which was a bit of a bummer because it was about extrasensory electroencephalic induction between identical twins. So it was published in Science, and the only figure was this. And so it was two ophthalmologists who recorded two twins and pretend to have demonstrated telepathy between twins. It has not been reproducible. But maybe that was what forced people to think that it was parapsychology in the first place. Because there are other papers in parapsychology who tried to use hyperscanning before it was a term. So after Montag coined the term and proposed that, there have been like several high-impact papers in neuroeconomics, especially, for instance, the Kinkasas paper of 2005, where they use economic exchange between people to investigate how brain activity of one person can help predicting the decision of the other. And over those 5 years, There's been like multiple fMRI studies more or less in this trend, but first things that we have to keep in mind is that it's not always hyperscanning between two people simultaneously. There are a lot of papers that actually piggyback the term in a way by using the concept of more ecological recordings and to look at interpersonal relationships, not necessarily by recording the two people simultaneously. And so, for instance, there is like examples of this very nice paper by Marlene Cheapers in Christian Kaiser's group, where they took like romantic couples and made a charade with gestures and scanning one person first and then another person after, and showing how the brain patterns of one brain can flow to another virtually through the communication. Another good example is the work of Urison in Princeton, where it's coined the term neurocinematics, like looking at brains of people while they're watching the same movie. So it didn't add like 20 fMRI in a cinema place. It's just like 20 people looking at the same movie, 20 times different moments. But interestingly, by looking at the synchronization and being physicist, I'm a bit picky about the term synchronization here because it's more correlation of bulk signal. But anyway, by looking at this correlation across people, what he envisioned is that there are peaks of similarity between people when they are watching ecological stimuli, and those peaks correspond to social scenes in the movie. So for instance, here in The Good, the Bad, and the Ugly. And interestingly, also with a very smart way of shuffling temporally the signal, it could also probe to what extent those, we call that time receptor, like receptor fields, but in time, you have a gradient of similarity between brains when you are watching a natural scene that goes from, of course, the sensory input, but up to very high-level structures such as frontal eye fields. So in this case, frontal eye field is also implicated in psychiatry, so it may also be a proxy for that. Meanwhile, other modalities, of course, started to do hyper-scanning because it was cheaper than having multiple fMRI scanners. So for instance, FNIRS, I'm not a specialist of FNIRS, but there are a lot of studies that have been done with FNIRS, a lot in neuroeconomic setting as well. At the temporal scale, it's also important to state that because of the dynamics of fMRI and FNIRS, the protocol design tends to be more like sequence-based for those techniques, while in the case of EEG, and I'm more from that community, people have tended to focus also on sensory-motor coupling and more real-time interaction. But it started first with the more hyper paper in the history of science with Hypermethods for EEG hyperscanning by Fabio Babilone and his team, where they recorded people playing card games, and they demonstrated basically that in the up to the gamma band, so gamma band is pretty fast, it's like 40 hertz, so it's kind of fast compared to the behavior. They show that on average, the patterning of activity between people in the same team were more similar than between people of different teams. So that was the first account that by using this multi-brain recording, you can tackle a bit this interpersonal sharing of information. On the same period, the team of Scott Kelso with Emmanuel Tognoli and Julien Lagarde also use hyperscanning EEG, but interestingly not for looking at interbrain relationship, but more to have intra-brain marker of interpersonal coordination. So it's like also a way to use hyperscanning for having a more ecological context, because as I explained before, like most of the study were focusing on one person isolated, passively perceiving social stimuli. In their case, it was more like having two people engage in active social coordination, and what would be the brain markers of this specific engagement at the interpersonal level. But interestingly, they reported in a poster at SFN in 2005. That's a very good reason to not skip poster session. I met also my wife in a poster session, so I've never skipped. But in poster session, you have like this kind of little results that are funny, but you don't know how to present them in a paper initially and so on. And so they were reporting idiosyncratic patterns of synchronization between brains. And, well, it stayed there. It's like a bottle, a message in a bottle for the community. And, well, later on in Berlin, the team of Lindenberger did it to prove it statistically speaking that there were interpersonal neural coupling. In that case, it was with dyads of guitarists playing the same song, but they have a non-parametric testing of those similarity of dynamics up to the EEG timescale. And, well, the point is that at that state of the literature, there were like proof thus that social perception of social stimuli in an ecological context lead to similarity of brain activity across people. But so it was social perception in that case, so it was passively looking at a movie still. The other piece of the puzzle was that EEG signals of multiple subjects can have similarity up to the millisecond scale, so that's more like the Babylonian work, but it was like on average on sequential interaction. There are neurodynamic marker of motor coordination, but that was intra-brain marker, and synchronization can be observed and quantified between brains, that the Lindenberger paper, but was potentially linked more to the external metronome and the fact to play the same song that leads to this similarity. So not at the endogenous level of the dyad. And so since I was interested in this inactive coupling between two people through perception action, we used a paradigm that was from developmental psychology initially, from dual, I call that behavioral-Hyperscanning. This was dual video with mother and infant. And the idea was to play with the condition, either to have real-time live stream of the mother and the child, or to have a pre-tape recorded video of the mother. And the point they show in this paper was that a baby at 3 months old is already able to decipher the difference. So he's able to capture this fine-grained dynamics of the contingency of social signals. And so Jacqueline was part of my PhD committee, and what we did is like reusing, we really took from the basement this old dual video system, and actually it's an interesting point from a methodological point of view, because now it's pretty hard to find analogic video. And for those experiments, if you are really interested in fine-grained dynamics, you need a super fast video system, dual video system. And analogic videos are still like the best for that. So we had like this very fancy optical coupled EEG system with like a video, analog video, all video analog. But, well, that was the point. And we had like those two people interacting via this dual video system, seeing the hands of each others. And we can play in the same way as the mother with the baby, either to have live or pre-recorded and adjust with contrast like that. So at the experimental design, and actually it's also interesting to present you that in more detail, because part of the pilot data that I have for the network are also this kind of design. So we can play with that, and if it trigger ideas for you at the modeling level, that could be the occasion to also think about it. But so we try to have a full gradient from uncoupled to fully coupled. So the ultimate uncoupled is everybody is on a separate room, and there is nothing on the screen, and we are doing nothing. So that's like a basic baseline. We call it no view, no motion, NVNM. Then you can have a first similarity at the behavioral level where the two people start moving their hands, but they don't see each other. So that's no view motion. And then we have a gradient from passive video watching of a video of hand movements, induced imitation, where one subject imitates the other subject in a forced way. And the last one was the most interesting for us, was the spontaneous imitation, where people can negotiate turn-taking with spontaneous imitation of the other. So of course it was balanced across the two blocks. And the idea, and that was one of the painful part, was that we discussed earlier about that. We are dealing with real-time social interaction, not with trials. I mean, small trials, like for instance in neuroeconomics exchange. So here you have to go from the behavior and extract your sequence for doing the neurodynamic analysis, which was actually painful. So like it took me 11 months to do frame-by-frame analysis of that. And I had to find another person to do that, to make a kappa-Cohen coefficient to see how stable it is. But so basically we were indexing frame-by-frame all the videos of the exchange, indicating imitation and synchrony. So imitation being defined as the morphological similarity of the behavior. So if someone do a movement and the other one do a movement but delay in time, it's still imitation. While synchrony is temporal contingency, not necessarily the same movement, but it has to start and end at the same time. So it's more like, for instance, you take a jazz band, they are doing different movement because they are playing different instruments, but they are all in the same rhythm, so to say. And so by indexing those two things, you have a gradient coming from synchronous imitation, where people do the same thing at the same time, to no imitation and no synchrony, where they are moving differently and not necessarily in the same pace. That's annoying. It's going to be faster, but so that's the nightmare software I had to use to index frame by frame. And you had like the two video of the people, and you index the different thing. Up. And in summary, what we found is that, well, this was the point of the experiment. Most of the time, people were spontaneously synchronized and imitating each other. And in second, it was more synchrony. So like a quarter of the time, they were doing different movement, but still playing the same pace. And there are very nice papers by Isartel called Can We March to the Beat of Our Own Drum? That's showing that it's actually super difficult to not synchronize with others, even if you are explicitly told to not do. So we are entrained by the behavior of others. And then imitation and no imitation, no synchrony, which was like 8% of the time. Interestingly now, we realize that these moments of absence of imitation, absence of synchrony, was also the moment of turn-taking. So it may have also a social dimension by doing so. So before going to the inter-brain analysis, we have also intra-brain analysis, because since recording two people in spontaneous interaction was not that common, we wanted to look at also, like Tonioli did, if there were like specific neuromarker characterizing that. So we used a classical power analysis, but using also cluster-based statistics. So that's maybe one of the things we can discuss in the workshop and zone after the brain vision stuff. But the idea is that you can make your statistic more robust by integrating priors about proximity of sensors or proximity in spectral domain of the effects you're observing. It's also working in fMRI, but here we did that to contrast different conditions of our design to extract, for instance, markers of self-agency, or we attribute the agency of the action, ongoing action, to ourselves or to the other, and also to understand if there were like markers of co-ownership of action. So, for instance, when you are really negotiating the action compared to how you are passively reproducing an action that the other is producing. So interestingly, we show that beyond the difference of social perception and social interaction, that was already a big case, there were also a modulation by the role you are taking in interaction and by the context the interaction unfolds. So basically, when you are interacting with someone else in a forced unidirectional way, your role in the interaction is settled more and tends to have more stable markers. While when you are in a spontaneous turn-taking based interaction, you have markers that seems symmetrical between the two people, even if they are not playing the same role. You still have identified marker of the imitator and the model, but you have stuff that you don't see in the induced case that are symmetrical in that case. And we're going to see with the interbrain synchrony that actually those symmetrical patterns in the right parietal lobe actually corresponds to where you find the most similarity at the neurodynamic level. Interestingly, with the team of Philippe Fossati and Sylvie Guillonnet, we reproduced these patterns in fMRI, not by having hyper-scanning, but fake hyper-scanning. So there were like one person that was recorded in the fMRI, and the other person was outside and playing spontaneous imitation. And they found also that there were like more symmetry between model and imitator in the spontaneous context, while there is this symmetry breaking in the induced context. And in their case, thanks to the spatial resolution and the ability to see more deeper sources of cortical activity, they show that this difference is mostly due to a reward system. So for instance, like being engaged in a forced communication like you are right now after your digestion is less rewarding than being spontaneously interacting with someone else. So I hope you're not too bored and not too... but so like that's the kind of takeoffs. Actually, that has implication for education, and we wrote about that for instance, peer learning would be more rewarding for kids, for instance, than traditional one-way teacher-to-kid teaching. So at the brain analysis level, one way to deal with that, and but we're going to see other metrics that are interesting, is to extract phase of the signals and look at phase synchrony. So I was lucky to be in the former lab of Francisco Varela, who used that for analyzing intra-brain phase locking value and correlate with access to awareness. But here, the idea was exactly the same. So taking the EEG signal, filter them in the relevant physiological band of the spectrum. Then with the Hilbert transform this signal oscillation into analytic signal where you can extract the phase of the signal, so where you are currently in the cycle of the oscillation. And by extracting this phase, you can actually, by taking two sensors, look at how the phase evolve similarly or not between those two sensors. And interestingly, you have a kind of dual decomposition. You have also the amplitude for the same price, so you can do also covariance of amplitude, which is another metric you can use for looking at interbrain relationship. And so the phase looking value, that's how it works initially. You take the phase of your electrodes, you look at the difference between the two phases, and by taking the average in absolute value, you have a number between 0, where like it's totally decoherent, to 1, where the two signals are perfectly evolving in parallel in time. So the problem is that first, it's non-Gaussian as hell, so you need to be careful about what you're doing at the statistical level. And it's biased by spectral similarity. So if your two signals are more similar at the spectral level, it tends to inflate this phase-looking value. So that's something to have in mind. So here is a demonstration of things. You have the two EEG signals of two electrodes of two people in blue and red. And you have the index of behavioral synchrony above. You filter, you extract the phase, and then you compute the phase-looking value. And then you can see how much your coherence at the brain level corresponds to the coherence of the behavior. And it's important to note here that, again, behavioral synchrony doesn't mean that the two people are doing the same thing. They are doing the same pace. So it's not necessarily the same movement. So here is another video that's not working. I'm sorry about that. But so like you can see here how by doing that for all electrodes, so you have like all electrodes for subject 1, all electrodes for subject 2, and it's like battleship. You have like electrode 1 with electrode 5. Then you can have electrode 1 of subject 1 with electrode 1 of subject 2, and so on. So you have a global representation of all the similarity. This is what it looks like at the topological level. So you have the classical antero-posterior phase-locking value in the gamma band, for instance, intra-brain, but you have also this inter-brain synchronization. And that's the non-Gaussian distribution that you have to be careful with. Okay. And so to deal with that at the statistical level, we had the chance to have like people like Eric Maris and Robert Ossenfeld. So Ossenfeld is the guy behind FieldTrip, major MATLAB toolbox. And they did this wonderful paper in 2007, very smart way of including prior about your data to have more robust statistics. So the idea is, I don't think... Ah, pardon, il y a un curseur?
Oui, non, mais juste le...
Ah, ok, pardon. Désolé. Sorry, désolé. So here, for instance, I don't know if you see the cluster here, but the idea is if you toss a coin and you have the same phase over a long period of time, there is a great chance that it's a tweak coin. So if you integrate in time, the fact you have a statistical significant difference between two conditions, so here is evoked related potential, so it's in time, you can integrate the fact that from one sample to the next one in time, there is a relationship of neighborhood that is meaningful. And by integrating this information prior in time, you can have a correction for multiple comparison analysis that is less conservative than the classical Bonferroni statistics. And interestingly, this way of including the prior, they take this idea further in Eric Mason and Robert Ossenfeld. They show that you can apply that at the spatial domain level. So for instance, here on the right, you see that, for instance, having a statistical significant 3 electrodes that are neighbors seems more like an effect than 2 electrodes that are separated spatially. So the coherence of the neighboring electrodes is one prior you can have. Then you have the same idea with internal functional connectivity. So if you have bundles of functional connectivity that share the same beginning and end, it's also more likely that it's a robust effect. And so what I did is just extrapolate the same idea at the interbrain level to have these clustering statistics at the nonparametric level. And so that's give these results that were like the proof that indeed, in absence of an external metronome, through perception action, people can synchronize their brain activity. But interestingly, we found also in the gamma band this kind of similarity. And I remember Jean-Philippe Lachaud, who worked with Varela on phase-locking synchrony, was like, how is it even possible to get gamma band synchrony between 2 brains? I mean, the behavioral channel of perception action is not at 40 Hz. And so now we have computational model that shed a new light on that maybe that if in fact it's totally spurious at the gamma level. You have like the behavior that's correlated to the theta band. So there are a lot of results, including the work of Karim Gerbi in 2007, showing that the velocity of the movement is equivalent to the theta phase. And now there is a growing literature of cross-frequency coupling with the theta nesting the gamma. And so what we're observing in the gamma band seems like an indirect statistical coupling that is not directly through gamma, but through the theta. So that's one thing. As you may foresee, it was not easy to publish that stuff. So we had to prove that in another way, with a different approach. And that allowed to introduce another statistical technique that can be useful for the design of hyper-scanning experiment, which is basically a resampling approach. So you take your data at the behavioral level, you shuffle by keeping the same statistical distribution of your behavior, and you do the same contrast 1,000 times in the brain level, and you see how your real data is compared to this surrogate. So it's like a bootstrap in a way. And this allows us to show that indeed this cluster of synchronization, especially in the alpha band, was quite stable. So, and afterwards, indeed, it's interesting to see that this gamma is not really a direct coupling, because indeed at the surrogate level it did not survive. So only the alpha and the theta also survived. So this was more history, then now a bit of the hypes. So I have, like, for instance, one example of the hypes, being myself implicated in this paper. So for instance, when I tried to publish this first interbrain synchronization, I got refused 12 times. So like I had to rewrite my paper 12 times. It was like a bit frustrating, but like that's how science is. And so with this paper, with Pavel Goldstein, a friend now that we met at a conference like that, we managed to have a PNS out of this experiment, which we discussed at the beer after a workshop. So that show how it has evolved also at how the editor are taking those interbrain recordings. Here, what was interesting is to show that actually, even without the interbrain results, we demonstrated that interpersonal relationship at the affective level and through affective touch was able to reduce pain in the other. So that's, to me, the real big results of this paper. But we show also that this modulation of pain is correlated to the similarity of the brain activity. So another hype example is this Tech-lepathy, I call it. So there's like growing interest in brain-to-brain communication. Actually, for the anecdote, that was the code name of my PhD project, brain-to-brain communication. But we never used that in the paper. And so there is recently many bioengineering groups doing brain-to-brain communication, pretending to have achieved telepathy through technology by combining brain-computer interface and TMS. So, well, if I agree that it's interesting from a technical point of view, I mean, the science is not really clear here. Like, what is the question, and to what extent basically combining BCI that's known since the 50s with TMS that is known from the beginning of the century, is it really a breakthrough and so forth? But you can see that in MIT Review and all those outlets. So it's crazy how it can hype behind that. And so that's really two comments. Like, but do you need hyper-scanning? And that's the thing. I had to review so many papers where actually the hyper-scanning is just like a marketing stuff, and the question itself that tried to be answered doesn't need hyper-scanning. They just suffer for nothing because it's a lot of suffering to do all these setups at the technical level and so on. So the first thing to ask yourself is, do you need that for the question you are asking? So that's also something that's important. So for the hopes, so as I say, well, there is still one thing that has not been achieved for now, which is like interpersonal biofeedback, which was already in the paper of Montag in 2002. What has been one of the hope, it's still ongoing, but we had a first proof of concept, is to never again do frame-by-frame analysis of behavior. So now you can use this advance in machine learning to kind of automate these pipelines of analysis at the behavioral level. So here in pattern recognition, we did a paper with a funny extension of the concept of Granger causality with classification. So we try to predict the movement of one person based on the frames of the other, and vice versa. And by using this classification, we can predict which one was the leader and the follower in the imitation. So that could be a data-driven way of automating this frame-by-frame analysis. Another big step forward is to integrate developmental study into that, and it's super challenging. I'm now working, I have a platform with Hyper-scanning and in the child and adolescent hospital in Paris, and I must admit that working with adults and now switching to children, it's a pain. I mean, I love children, but it's super complicated, and for artifacts and for managing that, it's adding another level of complexity, and it has to be fun on top, so you have to make the experiment fun. But so, for instance, there is, for instance, Vicky Leong in Cambridge working on those hyperscanning recording in child and parents. There's also Stephanie Hill in Vienna doing that, and it's something that it's getting momentum. Linked to the neurofeedback at the dyadic level, a very interesting development is hyperstimulation. So until now, it was more like correlational to observe that similarity of the behavior was correlated with similarity of the brain. But if you stimulate simultaneously two brains and you show that actually stimulating in phase or anti-phase affect the social outcome, then you have a causal demonstration of inter-brain synchronization as having a functional effect. And so, for instance, Giacomo Novembre showed that indeed, by playing with transcranial alternative current stimulation, you can enhance perception-action coupling. And there are growing literature on that. So here it's also interesting. Giacomo made a very good job here to show how you have your real pairs of participants, and basically the first way of probing to what extent there is an effect in your data set